Hello, everyone. It's a pleasure to meet you here at this conference dedicated to alternative proteins. Hannelore, Daniel, and we would like to dive deeper into this exciting field, and we will focus on how processing impacts protein functionality. I will start with the processing part, and then I will hand over to Hanni, and she will focus on the nutritional aspects of plant proteins. So first of all, let's take a very quick look at how the market for plant-based alternative products has been developed. And we can see here the revenue for meat and milk substitutes, which is predicted to increase by 230% and 230 and 140% from 2019 to 2029. And this impressive growth shows a shift in consumer preferences towards more plant-based diets. Not only industry is engaged in this field, the scientific community is also very, very interested, and we can see here a strong increase in academic publications, in particular in the last couple of years. And the common interest of both science and industry underscores the potential of plant proteins, not only as alternatives to animal products, but as versatile, hopefully sustainable sources that can be used in other food sectors as well. So which raw materials can be used for the production of protein ingredients? And we can see here categories like oilseeds, cereals, tubers and roots, but also materials from the aquatic systems like macroalgae and duckweed. And all these raw materials differ in the chemical composition, but also ease of processing. And the question now is, which of these raw materials are particularly suitable for the production of protein ingredients? And this is what I would like to point out on my next slide. We can see here the results of a very nice study from Wageningen University. And on the left, you can see the proximate composition of individual raw materials, the proteins highlighted in green. And we see, for example, that microalgae like spirulina and duckweed have very impressive protein contents of up to 60%, while the commonly used legumes like soybean have comparatively lower levels. And this raises another question. Why are so many legume ingredients on the market, but only a few from algae? And one reason lies in the different processing behavior of the proteins. And this is what we can see on the right-hand side. The x-axis represents the yield, and the y-axis represents the protein purity. And we can see that the individual raw materials can be clustered into two groups: the leaf and algae material, here highlighted in green, and the seed material in blue. And if we want to obtain a protein isolate from microalgae with a very high protein content, huge processing effort is required, and this results in very low yields. If we aim for higher yields with algae, the protein purity tends to decrease. And seeds behave different. And this is because seeds contain storage proteins, whereas algae and leaf material mainly contain metabolic proteins like enzymes. And the storage proteins can be much easier be extracted than the metabolic proteins. So let's take a closer look at how the protein ingredients can be produced, taking legumes as an example. The process for sure starts with the seeds, and then in some cases the hulls are removed. If we have an oil-rich material like soybean, an oil extraction step can be included as well. And then the seeds are ground, and this is our base material for the protein isolation. And from here, different processing paths can be chosen depending on the desired end product quality. The simplest process is the sole grinding, the sole grinding, which results in flour. And an aqueous as well as dry fractionation technique can be applied to produce protein concentrate. And to obtain protein isolates, we can see that a lot of different processing steps are needed to achieve a very, very high protein purity. So now that we understand the general processing to obtain protein ingredients, let's look more closely at how the individual products differ in terms of chemical composition and technofunctionality. Starting with the flowers, they have the lowest protein content and, not surprisingly, the sensory characteristics of the raw material, as well as the technofunctional properties of the raw materials. And this makes flowers ideal for applications where a more whole food-like ingredient is preferred. Moving up to concentrates, the protein level increases and the technofunctionality is improved, but this depends on the ratio of proteins, starches, and dietary fibers. And the concentrates still contain some odors and flavors specific to the raw material. And finally, we have the isolates, the highly refined ones with the highest protein content. And we only have traces of dietary fibers and secondary plant metabolites, which leads to a more neutral taste and an enhanced technofunctionality, in particular for high protein applications. But as the protein content increases, so do the processing costs per kilogram. And this is also a very important factor to be considered. In addition to the valuable compounds, plant materials often contain anti-nutritive compounds. We have heard of that in the morning. Most of them belong to the secondary plant metabolite classes. And these compounds can be decreased by means of processing, but in some cases they can become even more concentrated. And this is what I would like to point out on my next slide, taking phytic acid as an example. In the bar chart on the left, you can see the phytic acid contents per gram protein isolate. In this case, it was mung bean isolate. And we can see that depending on the processing conditions, the phytic acid level can either increase by 200% or decrease by 45%. And the reason lies in the net charge of the proteins, which varies or which changes depending on the pH of the aqueous medium. The phytic acid is typically negatively charged, and under certain extraction and precipitation pH values and electrostatic interactions between the proteins and the phytic acid can occur, which leads to an enrichment of phytic acid in the final protein products. And this should underscore that we should understand and carefully control our processes to avoid rising levels of anti-nutrients. As a food technologist, I'm particularly interested in the technofunctional properties and the physicochemical properties of protein ingredients. And with this slide, I would like to show you how challenging this could be. On the left, we see the protein solubility of commercial soy, pea, and fava bean ingredients. And in particular, in the alkaline range, we can see a wide variety of protein solubility. And this is very similar to the technofunctional properties, taking the emulsifying capacity and the foaming activity as an example. Here we can see also wide ranges in the technofunctionality, even within the same raw material group. So when we talk about protein ingredients and their application in alternative products, it's not only about chemical composition and technofunctionality. So the texture, the nutrients, the color, the flavor, all must be right. The ingredients list or the number of additives should be short, and sustainability must also be taken into consideration. And these are a lot of requirements for these products to meet. So let's look more closely at how meat alternatives are produced. And in industry, usually two processes are used. This is the low moisture extrusion cooking and high moisture extrusion cooking. And both processes apply very high temperatures between 120 and 180 degrees centigrades. Low moisture extrusion cooking leads to expanded, porous, form-like structures, which are ideal for the application in burgers, for example. High moisture extrusion cooking is followed by a cooling phase, and this helps with the formation of the fiber structure that we need for the production of chicken breast-like products or schnitzel or nuggets or something like that. So how does extrusion influences the nutritional quality of the final products? And to answer this question, I found a very nice study which was published quite recently in Trends in Food Science and Technology. And the authors compared a huge range of alternative products and compared them with their animal-based counterparts. This is just an extract focusing on plant-based meat alternatives. And we can see that a direct comparison of plant-based meat alternatives and the meat products is very difficult in terms of energy, protein, total fat, as they vary tremendously in these categories. In terms of carbohydrates, sugars, and fibers, the plant-based meat alternatives usually contain higher levels, and this is very similar to that what Daryl presented before. The authors also found, very similar to your talk, Daryl, that cholesterol, zinc, selenium, pantothenic acid, and vitamin B12 levels are often lower in meat alternatives compared to the animal-based counterparts, while iron, calcium, and other nutrients were often higher. And this is because these components were added to the recipe to fortify the end products. So what else has to be considered when we talk about extrusion? The findings in literature are not conclusive. Yeah, there are some positive findings that show that the protein digestibility can improve, or that we can reduce insoluble fiber and increase soluble fibers. There are other effects that are not entirely clear, like, for example, the bioavailability of secondary plant metabolites. And last but not least, there are also some negative findings, like a decrease in thermolabile compounds. This is not surprisingly, but also the formation of lysine or alanine as a Maillard reaction product, and therefore a reduction in the indispensable amino acid lysine. However, it has to be mentioned that all of those studies, or most of those studies, have been conducted in vivo. And what happens in vivo is now Hanni's part.